Our next speaker is Takanori Nakane. You've heard his name a number of times already. Takanori's research focuses on high-resolution structure determination to understand chemical aspects of biology. Takanori studied medicine at Kyoto University in Japan, and after obtaining MD and medical license in 2010, he proceeded to do a PhD in X-ray crystallography at the Iwata group in Kyoto University. And he also obtained an MPhil in computational biology in Cambridge. From 2014, Takanori worked on serial femtosecond crystallography at the Nureki group in Tokyo University. And in 2017, he moved back to Cambridge to join the Reliant team at MRC-LMB. And Takanori will talk about single particle cryo-EM at atomic resolution. Looking forward to your talk.
Okay, thank you very much for introduction. Today I'm going to talk about single particle cryo-EM at atomic resolution, which we presented in Nature last year. So first, what is the atomic resolution? Unfortunately, the term atomic resolution has been abused, as pointed out in this ACTA crystallographic paper. Indeed, you can build an atomic model at 3 angstrom map, but this is only with the help of geometric restraint. So you are not actually observing individual atom per se. So in crystallography, there is a very famous and traditional criteria of atomic resolution, which is 1.2 angstrom. And this is called George-Sheldrick's criteria. But why do we high resolution? I often heard that many biologically oriented structural biologists say resolution of 3 angstrom or even 4 angstrom is enough. And I agree that to answer some biological questions, that is already good enough. But for me, my interest is in chemistry of proteins. To answer chemical questions requires higher resolution. When I say chemistry, it means, for example, the reaction mechanism of enzymes or how protein bind to ligand. To answer these questions in chemical sense, we need to see, for example, alternative conformations of side chains, or solvent water and ions, or hydrogen bond networks, protonation states of side chains, and also very accurate interatomic distances. For example, I showed a 2 map of photosystem 2, which is the enzyme responsible for photosynthesis. And this its reaction center called oxygen evolving complex consists of this very interesting shaped multi-metal cluster. And in this left map, which is around 3.5 angstrom, all these heavy metals merge into one big blob of density, and you cannot see anything. While on the right side, this is published this year, and this is now 1.9 angstrom. Now you can see individual atoms, and you can start to discuss chemistry. But if we can further improve this, then we can start with hydrogen, and this is very important to solve the long-standing problem in photosynthesis. For example, if this water or hydroxyl ion or peroxide or oxo, these things need identification of protonation state and also charge state. So that is why I want to improve cryo-EM resolution further and further. But what limits the resolution now? There are several reasons for signal loss, like envelope function or heterogeneity in the protein, or radiation damage or misalignment. While some can be recovered by better computational modeling, like CTF refining or Bayesian polishing, others are physical issue and cannot be compensated for by software. There are also many sources of noise, like imperfect detector DQE or ice, charging, or fundamental noise due to quantum nature of electron called shot noise, and inelastic scattering. In this research, we focus on 2 effects, envelope function in CTF and inelastic scattering. We worked in collaboration with some Fisher scientists, especially Abe Kotekia, and tried a new Krios with 3 new technologies. It consists of a new code fake and Selectris energy filter and new Falcon 4 detector. Now all of them became a product and you can buy it. And the product value is better than the prototype I used in this research. Okay, so first, Falcon 4 has similar DQE to Falcon 3, but it's run much faster. So in Falcon 3, you can collect about only 30 or 40 movies per hour. But in Falcon 4, you can easily go to hundreds of movies per hour. That means you can collect more particles and get higher resolution within given machine time. The Selectris is a new energy filter that is more stable than existing ones. So this higher stability means narrower slit is possible and practical than existing technologies. By removing inelastically scattered electron, you can get better contrast in the image. And to understand the benefit of code effect, you have to understand the chromatic aberration. Waves with different wavelengths, like color or energy, are bent differently by optical system or lens. In case of visible light, red light and blue light focused along at a different point. So this leads to the blurring of image. This example was taken from Wikipedia. The same thing happens for electrons. Although we often say electron at 300 kb or 200 kb, but not all electrons are exactly as the energy. So always some electrons are slightly higher energy or at lower energy. By using code effect, this energy spread is narrower. This means better envelope function in CTF. Up to around 2 angstrom, the difference is almost nothing. But if you go beyond 2 angstrom, the difference becomes very large, and you have many times more signals than traditional X-FEG. Also, because code effect are operated at lower temperature, it provides longer life. So it is actually cheaper in long term, but it needs higher vacuum and occasional chip flushing. To test this new microscope, we first use the aberration, our very favorite test sample for cryo-EM. It is recombinant mass aberration provided by Dr. Yana Dishawa in Tokyo University. We collected about 3,000 movies in about 1.5 days. The microscope was at that time the prototypes and running very slowly, but nowadays it can be collected in probably half days. And we got about 360 hundreds and thousand particles. And from this, we got 1.22 angstrom. And you can see individual atoms as separated blobs. To get to such a high resolution, every detail matters. Because even tiny errors in CTF parameters, such as the focus or beam width, can shift the CTF curve completely out of sync at high resolution and completely destroy high resolution signals. So you have to very carefully refine these parameters. So I applied per particle defocus refinement, per micrograph astigmatism refinement, and per dataset magnification anisotropy refinement, and up to fourth order aberrations for every few micrographs, using CTF refine developed by Jasenko-Jivanov in our group. We also applied Edward sphere correction algorithm developed by Chris Russo and Richard Henderson. And as you can see, by applying these corrections, you can get higher and higher resolution at given number of particles. And quite pleasingly, hydrogens are visible in a difference map calculated between observed map and a map calculated from model without hydrogen. So, and interestingly, those peaks for hydrogens are located a bit further from the position of protons. Initially, I thought this might be a pixel size error, but it was not. Electrons see electrostatic potential or Coulomb potential from both nuclei and electrons, not electron density as in X-ray crystallography. Because bonding electron of carbon and hydrogen are polarized, the difference peaks are not centered on the position of hydrogen nuclei. This fact is already well known in neutron crystallography, but it was very, and also for material science, but it was very nice to see this effect in protein single particle analysis as well. The Falcon 4 detector have a new movie format called EER, or electron event representation. In EER, it records all raw detector frames without fractionation, allowing reconstruction from individual detector frames, which means about 250 frames per second. And in other words, in this case, it corresponds to 0.1 electron per square angstrom per frame. And from this per frame reconstruction, as you can see, at early frames, you can see density for these carboxylate side chains. But as you accumulate electron damage, those get decarboxylated and it is gone. I don't have time to explain how to process EER in Lion, but it is now implemented in Lion 3.1 latest version. And please read this section in our help documentation. It explains all necessary fields, what you have to fill in. And another interesting thing about EER is that EER stores event location on very fine 4 times super resolution grid, or in other words, 16,000 and 16,000 mesh. So this is has very high spatial resolution. But of course, DQE at such high super resolution is very low, so there is actually not much gain. So in practice, you bin and you work in 2 times super resolution grid or in just physical pixel grid. But still, and this is enough in practice, and there is virtually no resolution difference. But the good thing is that in EER, you know the position of electron as a dot, while in typical MRC rendering from EPU for Falcon 3 and 4, each electron are convoluted with 3 by 3 pixel blobs. And this prevents noise beyond physical nitrate from aliasing back and slightly improve the DQE. But this makes the file very difficult to compress. And in practice, this doesn't improve resolution much. So by getting EER and then rendering electrons as a dot, you can further reduce the file size. And then one movie is less than 200 megabytes. So having a very small data size is very important, because nowadays people can collect easily 10,000 movies per day or so. So this is a nice way to get small data size. Anyway, going back to the high resolution work, you might say apophaking is just boring test sample. So we collaborated with Rad Adriescu in LMB and tested this new microscope on his membrane protein, that is GABA A receptor homopentamer. This is a very important drug target for some psychiatric diseases. And we obtained overall resolution of 1.7 angstrom, and for very well-ordered region, the local resolution is to about 1.6 angstrom. And at resolution, you can see very nice density for example, this glycosylation, and also for all other parts, these hydrogens were also visible, even at this 1.6 or 1.7-ish angstrom. So this is a very nice result. And we did several comparisons of different energy filter and those parameters, and the details are please look at our paper in the Nature. But this is not the end of the story. So there is still more room for improvement. For example, currently the Bayesian polishing is not yet perfect. So there is some room to do better movie handling, especially at initial few electrons, because these initial few electrons contain the least damage, the best information, but it is the most blurred because of very rapid initial movement. So if we can consider, for example, rotation of the particle during exposure or defocus change due to dooming, and we might be able to get better resolution. And also currently CTF refining does not take care of about sphere collection. So if we do about sphere aware CTF refinement, we can get better defocus parameters and we get better resolution. Dimitri Tegunov M proves such approach are feasible and beneficial. So it is probably worth implementing in Lion, the similar algorithms. And we are also working with Garib and Keitaro Yamashita in LMB for better modeling and refinement of high resolution structure. This includes time-dependent modeling on, for example, radiation damage, or modeling different maps simultaneously to follow time-dependent evolution conformation changes. For example, from time-resolved stopped flow or quench freeze experiments. And we are also interested in modeling charge and also better visualize hydrogens. And we are also working to better model symmetries. For example, this is a slide by Keitaro Yamashita presented in CCP4 study weekend earlier this year. Please look at his recordings for detail. For example, in Lion, when we apply symmetry, the map is symmetrized, so the model should be also symmetrized. However, oftentimes this is not treated properly. In EMDB, we often see the map containing all chains, but all chains have very slightly different conformation, while the map are already symmetrized. So this is not ideal. So in LEFMAC, you can apply NCS or non-crystallographic symmetry constraint. And in the model, you have only one chain, but this is internally expanded to whole assembly, and also this symmetry information is written to the MMC header file. And also, you have to take care of the many constraints due to symmetry. For example, when you define D factors anisotropically, because of the symmetry, for example, you cannot have this skewed B factor distribution for atoms. Because of symmetry, you have a less degree of freedom than atoms in orbital position. So LEFMAC now takes care of these effects properly. For example, atoms on the special position, for example, iron on the 4-fold axis in apophratin, or chloride ion on the 5-fold on ultraviolet, these are properly treated now. And to our pleasure, now we see many high resolution structures from other groups as well. For example, my former colleague in Tokyo did high resolution structure determination of select averaging. And using Krios G4 with K3 and GIF, they got 1.93 angstrom, and they improved this to 1.7 angstrom using the same scope as I used for this apophratin Java project. So this is very impressive because considering that streptavidin has only 52 kilodalton, very small protein, and how to align. And also, Radodanev again in Tokyo achieved 1.31 angstrom using Krios G4 with XFEG and K3. And also, Holger Stark group presented impressive 1.15 angstrom structure using his custom-made Krios equipped with monochromator and CC collector. And his result also supports the facts that improving the envelope function due to chromatic aberration and energy spread is important to getting highest resolution possible. And there are also many sub-2 angstrom dataset on Empire, including ribosome, urea, GPS, AAV, and connection, and so on. Finally, I want to acknowledge many people involved in this research. And I also want to thank Empire depositors, because I learned a lot by reprocessing dataset on Empire. And by doing this, I learned establish the best practice for data processing, and this is important to supporting other users. So, and all of our raw data are deposited on Empire, and I hope you guys also deposit your movies when your structure are published, so to help us future method developers and also students and postdocs who are learning data processing. And thank you very much.
Thank you very much, Takanori, for a really wonderful talk and very interesting for many people, I'm sure. There's quite a few questions in the Q&A. Yuri Shaban is asking, you've used 3 new technologies at once. How much benefit every one of them bring?
Okay, so I think for this high resolution work, I think the improvement of envelope function due to code effect is the most important. For other things, for example, if is energy filter at 5 or 10 eV better than 20 eV, I don't know. I initially plan to do these more comprehensive tests, but this called various mess called stopped all these experiments, and then nothing happened since then.
Okay, thank you. Alistair Burt is saying, great talk, Takanori. Would you mind explaining how you chose the group sizes for your beam tilt, astigmatism, higher order aberration refinements?
Okay, actually, this is the problem of the prototype scope. It is already fixed in the product Krios. But for this one, I noticed this is proto beam tilt, and this is resolution from every few hundred micrograph and those things. And then I noticed this beam tilt is changing a lot. So that is why I have to group individual, not individual movies, but every several hundred particles. But in practice, what I would suggest is to do this kind of analysis, and as long as this is fitting the time-dependent change nicely, I think that's okay. Because error in beam tilt, or very tiny error, say 0.1 millilabs, doesn't really matter unless you are going to 1.1 or 2 and so on. So for practical, in practice, you just try grouping every, say, 2,000, 3,000 particles and see if, and then make plot and see if it is nicely following the trend. I think that's enough.
Okay, thank you. TJ Regan is asking regarding the 4th order aberration correction every few micrographs. Does this mean that CS or pixel size is changing over time?
Actually not. For this one, the CS pixel size magnification does remain very stable. The fact that I did that this grouping is simply because I wanted to do the beam tilt, and then I grouped particles, then everything gets grouped, defined parts, such optics group along the way. So for tetrafold, you actually don't have to do in such small groups.
Okay, great. Another question, now it's from Jeffrey Woolard. Thanks for your talk. Do you think it would be possible to fit atomic models directly into particle stacks during refinement? Why do you think there haven't been more work in this area?
I know some people are working on this, but I don't really think that is useful or prove beneficial. But probably if the particle sets are very highly flexible, heterogeneous, probably you make several reconstructions, maps, using, for example, CryoDragon, the next talk, and then define atomic model against these reconstructed map. But when you define map, perhaps defining atomic model simultaneously against several maps might be interesting approach, and Garib's group are interested in such things.
Thank you. You have another question now from Otto Berninghausen. Do you think your fantastic results were also possible with a normal Selectris opposed to Selectris X?
I don't really know about the difference between Selectris and Selectris X, and probably it doesn't really matter because this is a comparison and you can see yes, this is XFEG, but 3, 5, 10 eV slits gave no difference. So I don't think going to very narrow slits is probably not necessary, but this test is only for Java receptor, and at say 1.2 something, there might be difference, but I haven't carried out such experiment carefully yet.
Okay, we're making some progress here. So Joseph Bartley is asking, hi. in rely on polishing our ER files analyzed as post hoc defined dose fractions, or can the dose fractions be used to determine trajectories which are average to individual frames?
Yeah, so this is explained in this page. So, but by default, when you first do motion correction, you decide how many raw frames to fractionate into one virtual frame. And polish use the same setting. But by using a script called rely on an ER trajectory handler.py, which is distributed together with rely on, you can make this finer, for example, change this to 8 frames per fraction and so on, and then use it for polishing. But in practice, doing such fine slicing did not improve resolution. This is perhaps probably because we are limited by how we model early frames. So if we implement, for example, rotation or zooming or those things, we might get more benefit by using finer slicing. But at the moment, going to very fine slicing simply leads to slower processing and huge memory requirements, and it is not beneficial at all right now.
Okay, thank you. And final question. This is for Marcus Gallagher-Jones. I was curious to hear your thoughts on refining charge states within cryo-EM maps. Given the profiles of charge form factors for electrons, do you think the low resolution frequencies are sampled well enough in cryo-EM?
That is a very interesting approach, and I am also very interested to do this with both single particle and also with micro-EG. And yet again, I am working with Garib group on this topic, but at the moment, I don't know, how good the data set must be to stably define charge and those things. But if, of course, we can do this, it gives very much an insight to enzymology. So I am very keen to pursue this direction.
Perfect. Thank you very much for this wonderful talk. And I'm sure if people have more questions, please type it in the Q&A. And Takanori can answer it.
Thank you very much.
Yeah, thank you very much. And we, in the interest of time,